method development is arduous and often, I think, not as celebrated as it could be. Oftentimes, the applications that make those discoveries is what ends up being seen as most valuable. The enablement where you have to go through a crazy, tortuous journey. Once it's there, it's there. I think it partially gets taken for granted.
We're back.
Good to see you again.
Good to see you again. 5 minutes later. So, should we move to the next paper?
Let's do it.
Let's do it. The last one. To me, it is the most interesting one, and also the most challenging, I guess.
Mm-hmm. It is challenging even for the first author. There's just so much to it.
It's not any paper. So the title is Oblique Line Scan Illumination Enables Expensive, Accurate, and Sensitive Single Protein Measurements in Solution and Living Cells. And that was published in 2024. So recently, December 2024 in Nature Methods.
Mm-hmm, mm-hmm. Yeah, so I think before we dive into this, I need to give you a bit of context of why we even got there. So, ICON Therapeutics came about in September 2019. I joined ICON in April 2021 when the company, we had like 35 people or so. And something that we haven't talked about today is making measurements in living cells with methods known as single molecule tracking, as the name implies. You're tracking a single molecule over time. Mm-hmm. That's a method that I actually did a lot in my postdoc, a little bit in my PhD. And it's a method that has remained a relatively artisan one, where in one field of view in your camera, you're capturing one cell. You're capturing hundreds of, if not thousands of trajectories per cell with the goal of understanding how is your protein moving around as a function of the perturbations that you're throwing at it, as a function of mutations, And when I was a postdoc, just to give you some context, and this was at the University of Chicago, I would think it was a good work week. If I went home, I'm like, oh yeah, this week I captured 20 cells worth of that condition and 10 cells worth of that condition. What a productive week. Mm-hmm. Now, what we wanted to do at ICON needed to exceed that scale, because ICON wanted to take single molecule tracking and turn it to the point where you're capturing 50 cells per second, millions of trajectories. So you can, and now you're doing your imaging in a 384 well plate. So you can sample thousands of conditions per day, 10,000s of conditions per day, and do so with a level of statistical power that really allows you to understand, well, I'm not, it's not that I'm just increasing or decreasing protein dynamics. I'm now able to identify subpopulations. I'm able to identify very, very subtle effects, and it has to be robust. So the whole story behind this paper is, let's make single molecule tracking robust, industrialized, and powerful. And it all started with Eric Betzig again, who was a founder of ICON, who actually brought us the proof of concept for this oblique line scan, OLS. And our role and my role, working very closely with the research and engineering, well, mostly the engineering team, was to build a set of very specific requirements by which we were going to build these microscopes. And it started with one microscope, and eventually we built 30 of them. So at every part of the value chain, from the laser to the fiber optic to the lenses that create the light sheet, all the way to the cameras, all the way to the processing of the data, you had to like really break it into chunks and have specialists working on each part. And eventually what that gave them is a fleet of microscopes, where that were integrated into a robotic system that allows the experiments to be run automatically. So you can imagine you have a giant incubator where you can stick all these 384 well plates. There is a robotic arm that is interfacing between a variety of different instruments. There's an instrument that's responsible for adding compound and one for washing. And eventually they make their way to the microscope and the imaging starts. And, there are a lot of things that had to be figured out by a lot of people, but a very brilliant team that I worked with in the early days of ICON. So that was just some context before we dive into it.
Okay. This also, I'd like to talk about it. Like after the paper, I'd like to ask you questions about like spatial things that we can do, like spatial transcriptomics, spatial proteomics, because a lot of single cell. I'm very interested in single cell. I've been for many years working on for a few years now, and
sure.
if we can mix this kind of techniques with single cell to get basically a better image of what's happening inside the cells. But let's do that after. Let's talk about this paper first.
There's a lot to say there, but to be brief, I think what we're doing here is, we talked about turf, we talked about being able to change the inclination angle. What we're doing is you shape your laser into a very fine light sheet. Okay? And by shaping it into a very fine light sheet and then bouncing it on this thing called a galvanometer, you're scanning that light sheet really fast as a way to create the full sheet. Okay? Then what you're doing is that you're relying on the fact that on the emission side, you have a camera where you're relying on a rolling shutter. And you're not exposing all your pixels at the same time. So essentially on the excitation level, what you're achieving is that through the light sheet, you are basically reducing the contribution of out of focus elements. You're really exciting a thin layer. And then you're achieving better confocality. You're able to reject the background by getting your emission to coincide with the moment at which your camera is actually physically exposing those pixels. This was a tour de force that was non-trivial, that was achieved by my friend and colleague Martin Vanna, who's an electrical engineer. And I can tell you, it was pure wizardry to make that work very well in the early days. Like, just to give you context, this guy has been an electrical engineer since he was 4 years old. And it's been his hobby since. So, in essence, you shape the light sheet, and then it's being scanned really fast. You're able to scan a really large field of view, pretty much a good chunk of a large chip, within 10 milliseconds. And in that large field of view now, what you end up having, and I think panel E shows that quite well. Panel C, if you zoom in.
C. Understand. So very quickly, what do you mean by light sheet?
What I mean by that is that instead of just having just light coming through unshaped from your excitation going through your objective, what you've done in the illumination part of that gets into your fiber optic is that you've used something known as a parallel lens as a way to turn that initial ray of light into something that's a bit more defined, into something that's a flatter, nicely defined beam.
It's basically a more controlled beam over a specific surface.
And the whole world of light sheet has plenty. There are Bessel beams, there's this and that, and there are a few tricks that you can play to define an actual light sheet.
Okay. Thank you. So this is... yes?
Yeah, as you can see, That whole field of view, you have cells, you have nuclei in them, and that alone was impressive. You're able to just get 50 cells in a field of view or so, and then capture all the trajectories corresponding to each cell. And on the right, you have a zoomed-in version for one cell, where you can see what 10 milliseconds of imaging gives you.
Wow. So these are cells. So the borders that we see, they're basically it's cells, and the dashed lines. What do they represent?
Those are the nuclei.
Those are the nuclei, and the dashed square is basically what we see zoomed in on the other panel.
Yeah. That's right. And then you have the trajectories that are color coded as a function of how fast the particle is moving. So you have the diffusion coefficient per trajectory. And that, I can tell you, that is a game changer. Now I'm doing 1 week of my postdoc in 10 milliseconds.
That's a big difference. Wow. So so the blue is basically where it starts, and the 6, like the red, is the end of where that particle moved.
Not necessarily. So you have a lot of trajectories going in and out of focus, so you end up losing them during the acquisition. So you're capturing, so I'm wrong actually, I should have said, so it's 10 milliseconds per image, and you're capturing 150 frames. So it's about 1.5 seconds to generate this image.
Okay.
And so what you're doing is every 10 milliseconds, you have a still image of very sparse spots. And then at T0, T1, you're linking them to each other.
Okay.
And to generate these trajectories and the whole linking problem has been one that was open for a long time.
Yeah.
And one of my close colleagues, probably one of the smartest person I've worked with, this guy called Alec Heckert, very good friend, was able to deal with this optimization problem. It was a very intense, probabilistic, mathematical optimization problem of linking spots to each other one frame after another.
Yeah, it's not trivial. It's not trivial to follow the particles across different images.
Yeah, so I can tell you the details, but the guy was on an island alone, having optimized this.
He went into a retreat by himself to find a solution.
Yeah, exceptional guy.
Wow. You know what I find is I really like, there's another thing that I really like about these pictures is that they show you how full actually cells are.
They don't even show you 1/1,000,000th of how full it is.
Yeah,
now, that's one protein. Now layer on top of this the whole proteome moving around.
Plus the RNAs and plus all the other stuff.
It's an extremely tight, busy place where things are bouncing off each other just because.
Yeah.
And then evolution has optimized things for certain interactions to be more obligate than others, stronger than others, and that's how we happen to have biology.
Yes. Yeah. So imagine this is like a single protein. Imagine the fullness of a cell and all these things interacting with each other, and us trying to figure out basically what's happening.
Us as biology with trajectories happening inside us, trying to understand this.
Yes.
The biology measuring biology is not a trivial concept. So what you'll see in that panel next to this, there's that orange figure, D. So that little box shows you what kind of images we used to capture before transitioning to oblique line scan. So it's like 6 times more field of view. But not only more size, it comes with more homogeneity in the signal to noise ratio. So that is a map that shows you that as you're sampling through, your results are not going to be biased by differences in signal to noise ratio. Meaning, if you look at that square box and you look at the violet region, which is a lower signal to noise ratio region, if something is moving fast, you're going to have more trouble actually tracking that, not even tracking, identifying that spot, detecting that spot. So if you do not have something homogeneous, you're at the mercy of the heterogeneity in SNR, and you're gonna miss some things or skew your measurement towards the slower moving particles. So we increased it by 6x. We made sure it's homogeneous. So we know that the measurement is robust at the single field of view level.
Okay.
And then, since I told you we had to build a fleet of microscopes, what we had to make sure of, if you look at panel E.
So you have 4 microscopes.
Here we're just benchmarking this thing on 4 microscopes. I could have easily gathered data from 12. Well, actually at the time we didn't have that much, but we just ran the exact same experiment. We did this 20-point dose response. So what we took advantage of here is that there's this protein known as KEAP1 that heterodimerizes with this other protein called NERV2.
Yep.
And there happens to be a very good molecule that abrogates that interaction. It breaks the two up. And KEAP1 goes from being a heterodimer to a monomer. And it works really well. So then, as you can see, as you increase the concentration, you see the diffusion coefficient increasing. And what we wanted to make sure of as we brought these microscopes to life is that the reported EC50, the 50% response, was at least within threefold between each and every microscope, with the Emax hopefully being also overlapped. And, whenever I look at this plot, even to this day, it's just impressive that we're able to get that level of reproducibility.
Okay, well, so just to make sure I understood correctly, so you have two proteins that fix themselves to each other, and you separate them and with different concentrations, basically, and then you measuring the separation with different microscopes. And what you're benchmarking here is that your method is actually reproducible across microscopes.
And you're doing this with each data point for each concentration being a lot of different wells. So you have a lot of reproducibility of repeats per condition.
Yes.
And you've also randomized the distribution of the concentrations in your plate. You have not organized them in a row or a column format. You've actually randomized them as a way to account for potential spatial biases that may be introduced by where it is that you're making your measurement on the plate.
And DMSO being basically the negative control, it's the baseline.
Yeah. It's the solvent and it's the baseline.
Yeah, it's the solvent.
Yeah.
And then you add the different concentrations on different plates.
Yeah.
This is why, that's why you have like the error bars, because you have several plates.
Yeah. And those are standard deviations. Yeah. So it's even great that we're able to get something that tight.
Yeah, it's super tight. Nice.
And what makes me happy, and we can talk about it at the end, is that the technology is getting traction even outside of the walls of ICON. People are building them now in their own labs.
Nice.
So here it is just to show that we can change the frame rate. So you have this full field of view. If you start cutting the size of the field of view, you can now
that's F, right?
What's that?
That's F.
Yeah, F. Yeah. So you can image at 100 hertz, 200 hertz, and you can make it faster, but the cost is that you're making your field of view smaller. But by doing so, you're going back to the same region faster.
Yeah.
And the reason why that became important to us, you would say, well, you've bothered to make your field of view bigger. You're talking to me about a bigger field of view. Why are we going to make it smaller? The idea is, depending on how fast your protein is moving, you want to have a sampling rate that is appropriate to the fastest diffusing subpopulation, if any, of the protein that you're interested in. So if you look at the plot on the left of it, and under the dose response, you can see that for track length, you're optimizing for it here at 800 hertz. But there is another plot, I believe, that shows us that for this very specific example, the point of diminishing return in improving temporal resolution is about 400 hertz. There are going to be other proteins that might be even faster, where you may have to really pay the price of having smaller FOVs for the sake of improving temporal resolution, if the cost is just acquiring more images. Acquiring more images comes at a cost also.
I see. So for this specific protein, the sweet spot seems to be 800 images per second.
You see it in another plot that has to do with how you're recovering the subpopulation, but it's between 400 and 800, yes.
Okay. That's way higher than I expected. So you need 800 hertz just to see basically what's happening for the diffusion of that specific protein. So like it's really happening really fast.
To capture that fast-moving fraction really well. Yeah.
Wow.
To not be undersampling it.
So cells are not only full, but they're also moving really fast.
And to go back to some of the founding fathers of ICON, like so there's Eric Betzig, there's also Xavier Darzac and Robert Tijan. So Tej has been working on transcription factor biology for a very long time, and he identified the first eukaryotic transcription factor. And they figure out the parts to the machine, all the different cofactors and whatnot. And through his alliance with Xavier at around 2010, 2015, I don't know the exact date, and using this kind of methods, they went from models where they thought that transcription factors hang out around DNA for minutes or seconds, to then understanding that it's a very fleeting moment, that interaction, that is actually in the picosecond, microsecond range.
Wow. So just so I understand correctly, so transcription factors, they just around, but then the interaction is like a burst and then they stay around?
Yeah. And we don't understand everything about it. And I think the real experts are Tej and Xavier about this, but it appears that what we thought to be something where, well, it's just moving around and now it's sitting there to do its thing is a very transient and weak process. And, there seems to be a search mechanism that has to do with how compact DNA is, that has to do with heterochromatin, euchromatin. It seems to be that intrinsically disordered regions on some of the transcription factors actually play a role to identify the regions that they're supposed to bind to. At the end of the day, that's when I was saying earlier, words. When you say a transcription factor is recruited, no one is recruited. The transcription factor doesn't know what it's doing. It's just chemistry at its core, You
mean there's no one going around the cell and then recruiting transcription factors?
Yeah.
Like sending CVs.
And at the end of the day, it appears to be, when you look at these things, that it's just touch and go, and then the thing is moving around, just doing its thing.
Wow!
And there's so much to explore there using these methods.
Wow! That's wow! And just so people know, transcription factors are basically what define what if genes are going to be expressed or not.
Yeah, they bind to the promoter region and they activate a gene.
Yeah,
and
super important.
They do it as part of a large complex of a lot of players, and we never really understand the molecular dance of how those players come about and whatnot, but the players have been identified.
That's super important. I'm discovering so many new things because for me, when I work with transcription factors, they're basically fixed because I work with, for example, if you work with the genome, transcription factors in genome is basically a picture of the genome and that's it. So I never really think about the kinetics of the interactions. And I wasn't expecting it to be so transient.
Yeah. And I think it's only now that we're starting to have the tool to understand how a lot of biology is governed through a lot of very fine, weak, transient interactions.
Well,
it's changing the models completely.
Yeah, completely. It's fascinating. It ties back to what we were saying before at the beginning, the changing models. I would never have expected that the interaction of transcription factors would be so weak.
Yeah.
Wow.
So we can go to the next figure if you'd like, to give an example maybe of why this is useful. So here, what we did, we looked at this protein called VCP. VCP is linked to, what is it not linked to? It's linked to so many processes inside the cell. And depending on its localization, it seems to have different cofactors. If it's in the mitochondria, it's linked to XYZ. If it's in the nucleus, it's linked to ABC.
Yes.
So what we wanted to do was get a sense of, are we good enough at being able to see spatially dependent changes in diffusion coefficient?
Mm-hmm.
So basically, the only thing to really pay attention to here is you have an image on the left that has a non-targeting control, one in the middle that has a small molecule that's known to interact with the inhibit VCP across the cell, and then you have a knockdown, an siRNA knockdown of a cofactor known as Faf2. And that Faf2, if I remember correctly, is a cofactor to VCP only in the endoplasmic reticulum.
Mm-hmm.
So what we're demonstrating here is, okay, you have your overall diffusion where it seems to be going faster in the cytoplasm with a non-targeting control. The CB5083 seems to overall reduce the dynamics in a non-discriminating way across the cell overall. Mm-hmm. But then Faf2 is really impacting the VCP where it should impact it, which is around the cell in that perinuclear region. And we had other examples of this, but it was to show that we have enough statistical power to be able to look at these very subtle spatial effects.
Mm-hmm. Mm-hmm. Mm-hmm.
And that matters a lot. That matters a lot because if you're targeting a protein of interest, maybe you just want to target a subset of it. Maybe you want to target a subset of it that's part of that complex, not part of that other complex. Who knows? So do you have the ability to resolve that? I think we started getting a sense that it was possible.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
And I can see many applications of these kind of things. For example, for cell-cell interactions, you want to see what's happening, where are basically the molecules doing the signaling, what they do, and the pathways, how for example, the pathways would change what is happening inside the cell when a pathway is triggered. Where does this protein move? That's because so far, like most of these discoveries have been made basically through knockouts, maybe through phosphorescence with Brett and techniques like that, but mostly through knockouts and theory. But now we can clearly see, like we can image where the interactions are happening. So if we have a pathways of genes like a cascade, we can in theory know where each step of the pathway is happening inside the cell.
Yeah. And one thing that I wanted to highlight is, I'm here talking about single molecule tracking, but a key for these projects to work is to have the right cell lines. So again, I was lucky that we had a cell line engineering team that was generating the right cell lines, the right knock-ins, the right systems for us to make these measurements, because without that, we wouldn't have anything.
Yeah. And these are all primary cell lines?
What do you mean?
It means like did you make the cell lines or did you buy them?
we made them, but they were based on immortalized cell lines, U2S cell lines. They were not primary cells. That's what I wanted to clarify. Okay. But we engineered our own immortalized cell lines.
I see. That's super interesting. Just very quickly, like a knockout is it's just like knocking out the gene. It's like it's the crudest thing you can do to a gene. You just disable it.
It's you disable it, and it's risky in many ways, depending on how much of a central hub that protein is, how ubiquitous it is. Some of them are going to kill your cells. Others might be okay. And that's why with knockdowns, I think you tend to have a tool that at least allows you to modulate some of it, reduce quantities without completely preventing that protein from being there.
Yes. Yes, exactly. Because if you completely disable a gene, you first of all, you basically knocking out the gene, and we know now that genes are connected to many other genes. So it's not like the primary function of the gene that has been discovered is usually not the only function of that gene. Yeah. So if you knock out the gene, you're deregulating usually a lot of things in the cell that you don't even know about.
Yep.
So it makes it a very, very crude instrument. It was, for a very long time, it was the only instrument. But now, obviously, we're moving beyond that, and it's fascinating.
I mean, knockouts have been very essential in figuring out correlations.
Yes,
and
of course.
Yeah.
They still have their place. But it's harder to get a definitive conclusion for a knockout alone. Like, for example, an image is like, if we were to use a metaphor, imagine you're driving and it's raining and someone knocks out your wipers.
Mm-hmm.
It doesn't mean that wipers are fundamental things for a car to work.
Mm-hmm. Mm-hmm.
That if you wipe out, like in every condition, if you knock out the wipers, you increasing the chance of having an accident. That's not exactly what happened. It also depended on the context. Good. So.
So the other thing, I don't think we need to linger on this one too long. This is some work done by another colleague of mine whom I appreciate really much, Mason Bretan. Just a quick tangent. Him and Alec Hecker that I mentioned earlier. Mm-hmm. When you meet people who are like just truly on top of their game and they elevate your game.
Those collaborators.
These were just exceptional people at every level. Mm-hmm. But with what we did with Mason is that we were able to just, in collaboration with other people in the research team, just be able to identify stages of the cell cycle. Something that we show here as a proof of concept with this protein called PCNA that's just more present during replication. Mm-hmm. And really, this is all to say we can layer on top of our measurement, if we want, cell cycle. So that you can understand how your protein is regulated as a function of cell cycle regulation, which may matter a lot in the case of transcription factor or other things.
Mm-hmm. So this is what we're seeing here with the G1, G2, and S phase.
Yeah, that's right.
Phases of the cell cycle, which is basically cell division.
And it shows you that things are moving faster in G1, G2, and it's getting slower during S phase as the protein forms like this punctate.
Mm-hmm. There's so many things we could do with that in immunology and cancer. It's like so many things. Cycling T cells. Just trying to understand what is happening inside cycling T cells and what these ones are cycling and the ones are not cycling.
Now, with your T cells, the challenge you would have is you would need to find a way to image them or to get them to adhere, to be closer to the glass.
Yes, that's we probably should get a molecular biologist in the discussion because because the thing is, we cannot do that inside a living organism. Uh-huh. So it means that we need to create a model on basically in a petri dish, like in a glass environment, so that we can do this kind of imaging. And that becomes challenging in itself. That's in itself, like this is because it's the technique is amazing, but for that type of applications, we also need to create a model that's representative of the real biological phenomena.
That's right. Yeah. That's right. Yeah, maybe you and I should work on that. Figure that one out.
I would love that. I would love that. we basically already have a project trying to figure out which population of cycling T cells are creating some autoimmune disorders in cancer therapy. Mm-hmm. That would be, I think that would be a good application for that because we're still, it's like we're seeing it from a gene expression, mostly from a gene expression point of view. So we see with gene are being mostly expressed inside the cells and we can cluster them and look at that. But this gives us like a much higher resolution. It's not, we still get the gene expression, but now we can see the impact of the interactions inside the cells.
Yeah.
That's super cool.
And we can try to build the first one of these microscopes in Morocco. And one thing that is going to be challenging is just the quantity of data that you're going to be generating. I mean, it's not going to be icon scale where you have 30 microscopes times 400 plates a week, whatever it is. But the data management part is non-trivial.
Yes, definitely. Especially if we're dealing with cancer, because we need people. I mean, we can still, there's other ways. But the most interesting, meaningful type of research you can do is when you get patient samples.
Primary cells, hard to work with, hard to engineer.
Yes.
But that's going to give you something translational.
Exactly. That's the closest you can get to translational medicine.
So, your immortalized cells are going to be good for mechanism deconvolution in a well-controlled force system.
Yes.
But it still remains like, it's the 10th derivative to the system you're trying to drug.
Mm-hmm. Mm-hmm.
And that's probably a euphemism about the complexity that you're trying to face at the patient level.
Mm-hmm. Yeah, it's the complexity is always something we try not to think about it too much, just because of it's basically like limitations. It's for some things you cannot always, as you said, primary cells are very complicated. First, you need to get them, like primary cells from patients. First, you need to get them, which means you need to define your cohorts, ethics, all that, and for some diseases or I mean, there's just not enough patient to allow you to gather that like very quickly. So you have to wait to gather all that, which means you need a biobank to be able to gather the cells. So it becomes like really like challenging logistically. That's why people are using mouse models or cell line models, because you can iterate much faster.
Yeah.
But what you find on an animal model or a cell line, you never show 100% that it would translate to human beings, like clinically to human beings. So it's just basically a hack.
I agree. And I'm curious about your perspective on this as an AI expert. So there are a lot of initiatives going around here in the Bay Area trying to build what's known as a virtual cell model. You've probably seen that. Mm-hmm. And I remain skeptical in regards to these approaches, because it goes back to the exercise I think we mentioned earlier briefly. You need so many different types of measurements and for so many cross sections, such high quality data, to even have a chance at building a somewhat remotely useful model to model the complexity that you and I are talking about. So the effort, I think, needs to be taking place. But I think we are in the embryonic stages of it.
I agree 100%. I think the first thing is, so I see two major issues with that, with what's happening at the moment. The first one is we still don't have the data that gives a good representation of the complexity we're trying to tackle. But the first measure, like data just doesn't exist yet to the level of resolutions we want. So the best thing we can do is have models that would work well with the level of resolution we have, which is still could be very useful to a certain extent. But the other thing, I think the biggest issue is I'm convinced that the models, the type of AI models that we use at the moment, mostly transformer based models, have been extremely successful when it comes to NLP mostly, but also like videos and all that. I don't think they're adapted to any type of data, and especially when biological data. The first thing is, on a more general aspect, they are probably the equivalent of steam machines, which means they work, but they're pretty big, huge, and very costly to run. So we should probably be working on making them smaller, because this is it's basically like probably everybody has seen that picture of like like NASA, like the biggest computer that they have, like this computers would take rooms and rooms and rooms, and now you have more computing power inside your phone. So I think this is basically what transformers are. It's like this big stuff. Okay, they can do the job, but they're probably, in my opinion, they're highly inefficient. And the second thing is, which is super important, because efficiency is basically like energy consumptions, and the more energy you consume, and the more expensive you are, and which means the less you can use the models.
Mm-hmm.
So making them more effective in their energy consumption is probably like the number one thing we should be doing.
Uh-huh.
And I see very few people doing that. Like everybody's running with transformers, but like I would say, like let's try to find something better. And the other thing is, I think they're also in the way they used for NLP and image generation and all that. I think the data is so fundamentally different in biology that you cannot just like translate the use cases.
Uh-huh.
And one like the example that I like to give is if you take a book,
uh-huh.
and this is on protein structure, which is like what most people trying to apply AI is like the big thing at the moment. Like let's do more things with structures. It's like if you take a book and you have a typo inside that book, it remains the same book.
Yep.
But if you have a typo inside your protein, it becomes a completely different protein.
Uh-huh.
It can become completely non-functional.
Uh-huh.
Which means you cannot apply a transformer model the same way you apply it on LP to protein. And the same thing goes with diffusion models and all this type of stuff. You need to adapt it, or you need to find something better for biology, because otherwise what ends up happening, and I think this is something that a lot of these foundation models that people are trying to build are suffering from what I call a problem of underfitting due to overfitting. Like underfitting means you basically the model is not performing as good as it's supposed to perform. And overfitting basically means that the model has learned by heart, which means it hasn't generalized, which means it's basically a database, like a very expensive database. And nobody needs a very expensive database. It's a model that can tell you stuff on things that are not in the training set. This is what we call generalization. So you have, so basically what ends up happening is that this model will latch on some very specific things inside the data set, like for example this amino acid or this thing, and that will allow them to give you pretty good predictions on the training set, and stop learning basically. And when you apply them outside, and this is what people call the out of distribution problem, which is just another way of reframing the generalization problem. We already had a word for that. It's called generalization. So when you apply that outside of the distribution of the training set, you end up with a model that underfits. And still is very expensive. And this is what I see with all these attempts at foundation models so far. So I really think that before we apply basically quote unquote the same methods that we have applied to NLP that have been very successful. I think we should sit down and try to understand the biology better and the chemistry better, and then come up with models that are more efficient and more adapted to these types of measurements.
Yeah, I think you've summarized it perfectly, and thanks. That was extremely clear. And I think the danger, I always say, of overpromising based on the technological advancement. People are really smart. And the second you say we're 6 years away from curing all the cancers, people are paying attention.
Yes, they remember.
And now you've shot yourself in the foot, and you've made our life basically you only have so many shots at being credible and remaining credible. And I think in this world of misinformation and scientific elites having to justify their work, and we saw what has happened with COVID and blah, 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 blah. I think it's very important not to fall in hyperbole, because there is an amplifier or a technological accelerator. I think you have to remain really measured and humble in your communication. Yes,
I especially when you're doing AI. And the reason why I would say especially with AI, because AI had historically what we call AI winters. AI winters. I know that because I started when I started doing AI. It was during an AI winter, and AI winter is very simple. It's there's a breakthrough in AI, and then people overpromise because I think like the word is really ill chosen. Like AI, artificial intelligence, is like it fires up all kinds of things in people's imagination, and that creates a hype, and a lot of people are like hype is very contagious to a lot of people.
Yeah.
And they start building on that hype, that excitement, and then overpromising comes, and things are not delivered. And when you get high, after the high you get the low, and the low is usually lower than the high was. And
exactly.
This is what we call AI winters, and it means that nobody wants to hear about AI for a few years, at least.
And I think, we're gonna face similar things with CRISPR, with gene therapy, with CRISPR. I mean, you saw all the biotechs that have emerged wanting to do CRISPR-enabled gene therapy.
Yes.
I mean, you have a tool that looks promising. You have to figure out the hundreds or thousands of optimization bottlenecks for you to eventually potentially try to get something in the clinic. What is an off-target effect in your dish, which might be okay in your dish, takes on a different level of significance now that you're dealing with a patient, living for many years. And of course, everyone knows that. That's what I find very surprising. People are really capable and smart, but yet that optimization exercise and that bottleneck troubleshooting has not happened early enough. And the result is that a lot of companies have collapsed based on the promise of CRISPR-based gene therapy. What it does to the layman or to whoever is paying attention to it from a business perspective is like, this thing doesn't work. And then someone else is going to come with another technology, like this group that came with this print technology. They're like, haha, we have a way that, but no, you also have to go through the 1,000 hurdles of optimization before it becomes relevant. And
that's true.
You can't cut, there's no shortcut to it. And I think same thing happens with single molecule tracking here. There is like expectations. There is a promise by which you can do drug discovery better with these tools.
Yes.
Well, do you know the amount of troubleshooting we had to do internally? The amount of optimization that had to happen for it to even start becoming relevant, for us to have a drug discovery pipeline that can ride on this technology? It took, I had been doing similar work for 10 years. There were like 10 other people cut out of the same cloth. You had all the right players working 7 days a week, trying to get it to work, to eventually get relevance.
Yes.
So that's what this experience has made me realize. It's like, you need to build a freaking solid team to even have a chance, and you have to have enough time.
And enough runway.
Yeah, enough runway.
Money-wise, yes. And I would say that this is true for CRISPR, but it's even more true for AI, because the thing with AI is most people, and I think this is something that this is a realization that is lacking, and that really, really everybody should have, is that most of the AI hype, most of the people doing AI at the moment are basically sitting on the shoulders of giants.
Oh yeah.
Like really, which means they're using models that they don't know how to make, that have been developed using technologies that they don't know how to make, like not even PyTorch. Like most people won't be able to write PyTorch. And I mean, let alone designing. I mean, they haven't designed the models. They haven't designed the technology that's inside the models. They don't even know how it works. They haven't designed the thing that runs the models, and they're very, very far from understanding the hardware or just the administration of the whole system when it comes to high performance computing and parallelizing the GPUs and all that. And they either thinking a model that has been an open source models that are freely available on the internet that they don't fully understand, or they're using an API to a model that they understand less.
Yep.
And they're creating companies using that.
Yeah, it's nuts. And, being in the Bay Area,
yeah.
when I go to San Francisco, so I live in Berkeley, every 99% of billboard is some AI-enabled something. There's AI-enabled HR, AI-enabled accounting, AI-enabled sandwich making. But it's insane. And I think it hurts me even more I think it doesn't hurt me as much when it's AI-enabled sandwich making, but when it's like AI, there's a company, it's like, we're gonna disrupt biology or something. I'm like, stop trivializing something that you haven't even started to understand in the first place, for which you have no expertise. The work that has been done by tons of people over a long period of time, because at the end of the day, the incentives are not even the right ones for a lot of those companies. They are about profit generation and are trying to ride the hype bubble before it breaks. And I'll say it even here. I would say a lot of them are going to fall flat on their faces. A lot of the companies that are just slathering AI on everything.
Yeah. And coming from AI, the advantage I have is I started AI, like the deep learning, at the start of deep learning. So I saw the whole progress of all the things. So that gives me a very good idea of basically how these models work, because I've seen everything happening as I was, because I was at the beginning of the deep learning revolution.
Yeah.
And I can tell you from my personal experience, it took me 7 years to be able to sit in the same room as biologists and be able to talk in a way that they wouldn't notice that I'm not a biologist by training. That took me 7 years, and not 7 years doing it part-time, like 7 years full-time.
And I can tell you, as a biophysicist and a microscopist, it still takes me sometimes extra effort to sit in a room of true hardcore biologists or drug discoverers and make sure that I speak the same language as them, because my as you can see from the work that we've discussed, I'm not necessarily thinking about one protein, one pathway, and the outcomes of that.
Yes, exactly. And when I say I can do that, I can do it for very specific topics. Like immunology, I can do it with most of immunology, because I've been working almost on immunology for that. But for other aspects of biology, it's still out of my comfort zone. So yeah, and it's really, really complex. So that over delivering and saying we're going to solve biology and are we going to use AI agent for everything? First of all, you're not controlling the agents. Like almost everybody that's doing AI agents are wrappers over other LLMs that they're using through an API. So you have no control over that. It's they can decide to raise the prices. They can decide to change the model. They can decide to do pretty much anything, and your agent is dead. So that's the first thing. The second thing is I've seen a lot of overpromising all the time. I've seen it for image generation. I've seen it for code generation. Honestly, I use AI for coding less and less. When it started, I was this thing looks nice. Now I can see how I can basically it replaces a very junior employee. That's definitely the case. You can generate some. The code generated would be is higher quality than someone who's something that someone who's very junior would do, for example. But still, when we're talking about code, like real complex code, it's not capable of doing that. We're very far from being able to do it. And this is something that I'm very, very, let's say passionate about, because code to me, this is basically my whole life. I've almost been programming every day since I was 12. So coding is something it's personal. It gets personal. When someone tells me this is good code, I'm like, no, this is not good code. Yes, this is not good enough. And I have very clear reasons why it is not good enough, because there's so many different aspects that you have to keep in mind when you're programming something. It's the same thing with microscopy. Yes, when you're a certain level of technicality, you see so many different aspects. The code has to be readable. It has to be maintainable. It has to be secure. It has to be, and when I say maintainable, it means a very high standard, which means you can bring someone in the team 5 years from now.
Exactly.
And they would still be able to use it and improve on that code and understand it.
That's exactly it. When I was at Icon, I'd always say, I'm doing this so that when I can leave, people can still do this.
Yeah.
It shouldn't be person dependent. There should be such a tight turnkey system, a process, documentation, everything that you need so that you've taken it from the hands of, let's call me the expert or whatever it is, to now democratizing the tool. And once you've succeeded in democratizing the tool, to me, it's the biggest testament of, does it work? Does it truly work?
Exactly. Does it truly work, or is it just your hands that are magical? And that's super important. That's super important. And I think there's like that aspect of maintainability when it comes to code was already overlooked. And you can see it because a lot of companies, if you look at, so what you want to do when you're developing a software, you have developers. And the ideal case is that you want to have almost 100% of the developers developing new features. But when you look at what's happening inside companies, is that the vast majority of developers, after a certain point, they're not developing new features, they're just doing maintenance. They're fixing bugs. That's all they're doing. It's like this, the thing is so leaky that they fix something, they break 2 other things, and they have to hire 2 other people to fix the things. And it's that's why you can have like companies with 100 developers not shipping any new features for years. Because that's what they're only doing maintenance. And
I think you're completely right.
Yeah. So it means that aspect was already overlooked. Now we are at a point where it's with these new AI tools, it is even more overlooked. So I'm like, let's see what's going to happen 3 years from now with all these AI generated codes.
Might be a very nice house of cards that we're building. Probably.
I think, I think a lot of companies will have like very, very bad surprises in 2 years, 3 years from now, when they're like, oof, we probably have to scrap all this code and start fresh. And that's a very expensive decision.
I'm in the same opinion here. And, I think having strong fundamentals, whatever it is that you're doing, and having that breadth of knowledge to put it in context is going to be really important in the next few years. Dabbling in things, and applying the newest, shiniest tool, that's a risky approach.
Yes, I think you said it perfectly. Breadth of knowledge. I think this is what differentiates a junior from a senior. Someone who's senior has a breadth of knowledge, of perspective on what you're thinking, of basically the thing that you're building and the different interactions, like all the different aspects of it. And you get this very high level, basically high level definition of the thing that you cannot put into words. You can only give it somewhat to someone through mentorship, but you cannot never write a book that describes all the things that you have in your mind. And that's when you become a senior. When you're junior, it's like, I'll use the code analogy because that's what I know best. You could use a football analogy because, as we said before we started, or microscopy analogy. But for me, code is the natural thing, is that when you start coding and you bring a new developer in the team that's very junior, just got their diploma, basically they're just working on a small piece. They have no idea how that piece fits into the big thing. But as a senior, you see not only how this thing builds into the whole thing, you see also you have this temporal way of seeing the thing. It's like you see in the future
Yeah.
Years from now, how that piece of code will still be valuable and what it will be doing in the future. That's when you get really to that senior level. Before you get to that level, I don't think you're senior. No. You are working towards that, but until you can see it temporally with all the different interactions, I think you're not at that level, and AI is definitely not there. Like definitely.
Yeah. Well summarized. I think I agree with you. As you build your expertise and you enrich yourself with a lot of experiences, you build that knowledge base. And the reason why I said enrich yourself with a lot of experiences, there's even, I try to never decouple the professional growth from the interpersonal growth. And eventually I think you're in a place where, as you said, you can start piercing through the barrier of knowledge, and you can start having an intuition that is based on all the things you've done. And I'm emphasizing the word done. Because if you've truly done it and touched it with your hands and played with it and really wrestled it, I think you're in a position now to come up with the next thing or innovate. And I think, so what degree are you ever innovating or inventing? I barely know the answer. You look at GLP-1, for example. Some guys in 1906 in England mushed up some stomach and extracted the protein and realized that it had some relevance in regulating blood glucose. Then the whole thing went dormant forever. Then people were interested in injecting it in blood. The protein degraded extremely rapidly. What got us to eventually have this resurface was we realized that there is a lizard, I think it's called the Gila monster, that actually makes GLP-1 in a way where it degrades, it doesn't degrade as fast. So nature has an optimization space that is way bigger and has done so for a long time. Nature is a true inventor. And we've been able, I think, to learn a lot from it. Yes. And we've been able to create tools to measure it, to understand it. But I think that the invention or something, there's nothing out of the blue. Everything rests on esoteric observations that have been done since time immemorial, all the way to having in your hands a tool, Yeah.
Oh, yeah. And this makes me think of a very important concept. There's the concept of supply chain that's basically to space, but there's also a supply chain through time. And I think this is also like a fundamental aspect, like that supply chain from time. And I realized how important it is when I moved back to Morocco. Because in Morocco now we have pretty good coding schools, so we get very good juniors. But this is a new thing, which means we don't have that many good seniors, and that creates an interesting case, because if you look at other countries that had like this programmer schools, like before they were called developers or coders, called programmers, you have this basically this link of people. Like if you look, for example, in the United States, like programming has been done since the beginning of computer science, like modern computer science in the United States. So there's always someone more senior than you to teach you the stuff. But if you don't have this supply chain through time, it becomes really hard to develop it yourself. And this is something that's also very extremely relevant to science, because when like we're talking for almost like 3 hours about, now we're sitting in front of a paper that's you're imaging like single molecules and the kinetics of how they diffuse inside the cell. That's mind blowing. It's like, it's crazy stuff.
It's all related. I mean, regarding your point about
but this exactly, it's only possible because of that temporal chain.
I completely get your point, and be it in a cell or in a system, which might be a country, you have to have, in the case of, let's say, doing computer science in Morocco, the details better than me, but you need to have a precedent and an infrastructure and a space that will allow the things to grow in a way that's going to be satisfying. And I think you going back is a great thing.
Thank you. But it's true. And so I don't think there are problems in life. I think there are only challenges, and challenges are fun. So, this is a very interesting challenge. It's like, how do you bring basically a new, how do you solve that temporal supply chain problem is a very interesting problem. It's not just a problem in Morocco. I mean, so other countries, it's like the majority of countries on earth have the same problem.
Yeah.
And, and they reframe it differently. They say, oh, it's a development problem. I stopped thinking about the thing in development way, because I don't think there are developing countries and countries that are developed. I think all countries are developing, just at different rates and different ways. But I think this is a more interesting way of reframing the problem. It's like, we have this temporal challenge of supply chain, because we're just getting into doing this kind of stuff. How do we solve it? And historically, this has happened many times over. It's not like everybody finds the same technology all the time at the same time. It's like every country had to deal with that at a certain point.
Yeah.
But the thing is, I don't have enough historical. This is not something that is taught basically at school. So I'm still thinking about it. It's like, how did this technology move from that place to that place and got adopted, and then was able to fructify in that geographical region, and why it was indeed adopted in another region where they had the same opportunities.
Yeah. I think it's an interesting thing to zoom in for sure. I don't want to throw generalities, but for example, if we take this microscopy method, to me, the most valuable thing is when I hear from colleagues that I meet at conferences that they're building the same thing in their lab. Then what I'm hearing is that there's going to be an undergrad or a PhD student that's going to be doing that, and that's going to be running their experiments on that, who's then going to mentor another person. And and I don't even really care that it's my method. And now you're propagating an approach that then gets refined by others, an approach on which people can add on. And there has to be an exercise of knowledge dissemination that is better. I think that, a lot of these papers are behind a paywall. Unless you can get an older version, maybe on bioRxiv, if the authors have decided to do so. But we need to create an incentive system that is so much better so that information can flow better. Yes. And that we don't have the incentive at every freaking paper to be able to say in the discussion or conclusion section, we have determined the interaction between A and B. This holds a lot of promise towards cancer. Stop for a second, because that's going to, the Daily Mail and BBC News are going to pick up on that, and then people are going to read that. You just have to be able to share things for what they are. We looked at this, and it was a really weird observation, and we don't know what it means, but it's new. Could be something. We measured the something really well. Do what you want with it. I think there has to be more of an ecosystem that allows that, in addition to a publication system that is not the one we currently have right now. Most places do not have access to most things.
Yes, there's the publications. There's also mobility. Mobility is important for students from African countries are severely disadvantaged because of the visa situation.
Yeah.
I don't think people realize how severe the disadvantage can be if they haven't experienced it firsthand. But moving moving to just for a conference can be challenging.
Yep.
Just for 3 days. it can be challenging because of the visa situation. Plus the fact that usually the currency is pretty low. So, but fortunately, journals and conferences now are aware of that issue. So they
provide more travel grants.
Yes, they provide travel grants. They adapt the pricing. So that's very good. But the visa situation is still an ongoing situation. It's not easy for students to travel for conferences or travel for internship as easy as it would be for someone from the countries. And that's also very important because it prevents them from getting that mentorship that's available to other people around, people from other countries.
I think if you take someone, let's say, who's American or British, whatever it is, in many ways there is an inherent advantage of having a certain passport, and that already removes the fact that you have to deal with all these variables that you just talked about. So already your brain is freed up. You're using less RAM surviving. You're now focusing more on the intricacies of whatever research project you're working on. So having someone who has to deal with administration or immigration at the same time, even if they're excellent, there is part of their system that is consumed by that. And then I'm pretty sure that it plays a role. And then those that you see who have navigated immigration and were excelling, I think they did it through resilience, through a level of resilience that is rare.
Yeah, exactly. It is rare. And just to be clear, we're not even talking about immigrating necessarily to another country. We're just talking about spending 3 days coming back, and that those 3 days can cost you. Those 3 days can cost you months of work, 4 months of administrative burden.
That's right.
Is to spend 3 days at a conference.
Yeah. So going back on the paper,
yes,
I think we covered most of it and the essence of it. And here we're just showing that we can do the same thing in solution. So purified proteins.
Yes.
And if you go down, because the last figure is to say the final one here. And there are more figures in the extended figures, but this is just to say, and this is an open access paper, so people can access it. But you remember all the stuff we were talking about earlier, the storm and whatnot. We can also do it on this microscope with a larger field of view. And then we can do, in addition to single molecule tracking, something known as fluorescence recovery after photobleaching in the top right, where you bleach an area and you see your intensity come back as a function of time. And that was just to be able to say, look, we can correlate the single molecule tracking to the FRAP, and it serves as a control in a way. And it was just a little tour de force to say we don't have a one-trick pony microscope. We have something that's closer to a Swiss Army knife, and it's still compatible with a variety of advanced microscopy methods. And the rest of the paper is a lot of technical stuff that has to do with how the microscope was built and whatnot. But this was such a journey, and it got me to work with some of the best people I've ever worked with. So it was great.
It's a great paper. It's a fascinating paper. I would love to couple that with gene expression and other types of measurements. Really, I think the way I'm thinking about it, I'm like, if I could have this technology. And then, I don't know, any type of modalities, for example, single-cell transcriptomics, could be single-cell site-seq, which allows us to do surface protein plus gene expression. Or, for example, exosomes, sequencing the exosomes emitted by cells, immune cells or cancer cells. And it's it would be, in my opinion, it would be a game changer. Being able to work on that type of data plus the data that we have at the moment, that would be amazing.
And so what would be based on what you're seeing here, what would be the dream experiment?
I still don't know. I'm still thinking about it because I'm like, okay, we have these cells and they're interacting, and the interactions they have basically defines the phenotype of the disease we're trying to characterize.
And
so if you can have, for example, let's say you have, so one of the, I think the most interesting things in biology at the moment in that type of stuff is exosomes, extracellular vesicles. So you have cells emitting like small packets that have a payload that gets inside another cell and then changes the other cell internally. And so the thing is that if we can have, like, let's imagine you can measure these exosomes, and cancer cells emit exosomes, but also immune cells emit exosomes. So if you can have an idea of what happens inside the cell, like having a picture of what's happening inside the cell through time as the exosome enters the cell.
I get you. So I can almost already see the experiment. So it's one where, you would be doing some sort of time-lapse imaging over days, maybe, or hours at least. And then you as the event of exosome internal coming into another cell happens, you would now switch your acquisition method to be one that has a different temporal resolution. You want to go much faster, and you probably would have fluorescently labeled the things that you hypothesize to be of relation to whatever your biology of interest is, and now suddenly you're capturing these kind of measurements. So I think yes, it's doable technically. That would be
great. That would be great to
We should do that too. We've got 2 projects now.
Let's do that. And this is basically the new frontier. In my opinion, like the new frontier when it comes to single cell is new measurement. It's not foundation models yet. I mean, it's new measurements, new ways of combining. We're still at the phase where we need to invent new methods and combining the measurements of different methods so we can get like a more accurate picture of the cells.
Completely. And you need so many angles to look at the problem. And right now we're showing that the oblique line scan is really good for looking at cells on a dish. But now, I think earlier, 2 hours ago or so, we started the conversation with multiple cell types, tissues, organs. Like, you want to add complexity for it to be most biologically relevant, physiologically relevant, and eventually therapeutically relevant. And it's going to take time. And I look at it. I look, for example, at the work that Eric Betzig is doing in his lab. They've already made an improved version to oblique line scan. And they're throwing in their whole zebrafish or what have you, and they're looking at the whole volume. And it's only by persevering in that direction that you're going to be able to do something. That said, the incentive system barely rewards instrument building and methods building. It tends to reward a bit more of, we're a group that's discovery. Yeah. So I don't know how to solve that today, but...
I mean, it's the same with bioinformatics. It's, I think the best way is to, the way I'm tackling personally the problem is to do the discovery with the method. So you design the method and basically you do the discovery at the same time in the same paper.
Yeah.
I think it's probably the best way also to sell your method, because especially, I mean, in microscopy, it's a bit different, because the costs are usually way higher than in bioinformatics and AI. So, but in AI and bioinformatics, like especially AI at the moment, you can have an inflation of methods. This is what we have. It's like, there's a lot of methods being published in conferences and different journals. And, especially like if you put yourself in the shoes of a biologist that has a lab and maybe a bioinformatics team, and, they want to measure a specific thing, and they're the primary users of the method, which one are they going to pick? Because they all have benchmarks.
Which acronym out of all the acronyms available?
Yeah, which acronym? And they're not necessarily equipped to evaluate the methods themselves. So what they do is the most reasonable thing is use the method that we've been using so far that has been established.
Yeah.
So, and the best way, in my opinion, to convince people that your method is worth trying is not just to have the benchmarks, just something that you should do, but benchmarks are never never exhaustive. Because at the end of the day, you're always benchmarking on some specific cases. You can have you won't have 10,000 different benchmarks in your paper. That will never happen. So you're always benchmarking on some specific data sets, on some specific cell types. There's always a specificity there. So I think one of the best ways to convince people that your method is worth trying is to discover something with your method.
Yeah. Well said.
Yeah. And to do that, you need to understand something about the topic, about the biology. So, well, that means you can specialize, which is also nice. But you can hyper specialize in some specific things in biology, and then you basically start using your method becomes the way you make these discoveries, and because you make these discoveries, people are more interested into your
That's
method.
right. That's right.
Yeah,
agreed.
Mm-hmm.
All right, so we're done with this paper, I think. Do you want to discuss anything?
I think we're good. I think we've been talking for like 3 hours and more. So, right. Is there anything you'd like to add?
No. I just wanted to cover maybe in 2 minutes what I'm doing currently.
Yeah, that would be. Yeah.
So after ICON, I moved to this place called CxT Discovery. It's hosted on the UC Berkeley campus.
Are you
the
sure
founders are the same.
I'm still waiting for the sun to rise.
It's raining, actually.
Okay.
And the founders are the same founders of ICON. So I'm working with Xavier Darzac and Robert Tijan.
Mm-hmm.
And what we're trying to do is leveraging the PDB and AlphaFold tree, identify mutually exclusive interactions. So protein X and protein A and B interacting on the same exact interface. The idea there is creating inherently an amount of competition. And what we're trying to do is, if we find that there is a disease mutation at that interface, can we stabilize that using a molecular glue? But more than just the drug discovery component, using that database, looking for new interactions that may be leading to that competition and that may be leading to crosstalk between different functions. And to do that, I'm not using very sophisticated tools right now. I have a team of 2 research associates. We're using a lot of established assays, but we do have an OLS microscope that will be deployed at the right time at the right place. So that's what I'm focused on really at this point.
No. And how long have you been doing that?
For 1 year now, exactly.
1 year, so it's still pretty new.
Yeah, we just established the lab, brought the team in. We're still warming up. We're about to press the gas in 2026.
Okay. Well, that's very soon. I mean, this podcast will probably be released in 2026. We're in December. So it's probably we're at the end of December, so it will be released in 2026.
All right.
So that's okay. And are you planning on publishing a paper?
Yeah, we are aiming to put together a paper by the end of January. It's probably going to be published sometime in 2026. You know how that goes.
Yeah, so everything happens in 2026. Yeah.
So you'll get to see one of our model system that we've been working on. I'm presenting some of those results at the Biophysical Society conference in San Francisco in February as well.
Okay, that's nice. If we want to come back, we can do a second another podcast if we want to present the method. I think that would be. I think
nice.
Yeah. Because this has been the longest podcast so far. It has been a fascinating discussion.
Yeah, I need to start my vacation. This today was my first day of vacation. I'm supposed to be driving somewhere, so.
Yeah, but I mean, of course, not today. When you get the paper, when the paper is published. Yes. It would be nice to see the new things you've developed.
Sure, that would be a pleasure to talk to you again and hear more about what you've been up to.
Thank you, Amine.
Well, Tyler, thank you for having me and having this nice and long conversation. I think that was fruitful.
Yeah, thank you. It was a fascinating talk, and
right.
I wish you the best and best vacations possible.
Oh yeah,
because I know that you've been working. I know that you've been working very, very hard on everything that you presented. So
thank you. Now to you as well. And all I'm going to be doing is watching the African Cup of Nations.
we wish the best to the Moroccan team.
It's going to be tough. They're playing at home. They have a very good team, but at the end of the day, it's not the sum of the players that wins you the tournament. It's the team.
It's a team sport.
It's a team sport.
It's a team sport.
You look at South Africa. Their team is made of players that come from two teams in South Africa, and yet they beat us at the last African Cup 2-0.
We'll see what happens. The Moroccan team has been on a good strike so far. So let's see. We'll see. But this time it's, as you said, it's at home for the first time, I think.
No, in 1988.
No, 1988, yeah.
They made it to the semis. They lost to Cameroon in the semis.
Organized in Morocco is probably the first
1988 was in Morocco.
It was in Morocco?
Yeah.
You're teaching. I'm not very good at football. You're teaching me something here.
I can cover every African Cup all the way to 1976, I think.
Really?
But I remember all the details from the 90s and the 2000s. But the other ones, I have like literature knowledge.
So that's it's you're really into it.
Oh yeah.
It's like you were not joking at the beginning, like before we started recording. Really,
I'm gonna be doing nothing and watching the African Cup.
Good. And so I'm curious to see your analysis of the different matches. I mean, we can, I mean, it's interesting. I'm not really into football. That's not a secret. Like everybody who knows me knows that. It has never been my favorite sport. But obviously it's the Moroccan team playing. So, I know who I'm rooting for.
Yeah, no, it's going to be good. I think we just have to make sure, just like in everything we described today, to bring a nice conclusion, hopefully. That tournament always gives me the impression that it's about Brazilians. It's like, maybe it's a guy who's going to jump slightly higher and the guy who's ready to sprint and poke his toe at the ball in the last stretch was going to be able to either prevent that goal or put in that goal. The player is extremely technical. There are tactics. But eventually it's going to come down to who's ready to break his leg.
Yeah. Hopefully nobody breaks their leg.
Hopefully not.
Hopefully it doesn't happen. But I would say that, and I don't watch football very much, but one of my favorite competitions is definitely the African Cup.
Yeah.
Like definitely.
Yeah. It's fun.
It's one of the most fun competitions to watch.
You get some backflip celebrations after goals. It's all good.
Yeah, it's like the vibe is different. The vibe is different. The teams tend to take more risks than in other competitions. It's it's fun to watch. It's fun to watch.
All right. Well, with that, Tarek, thanks a lot again for having me, and talk to you soon.
Yeah.
Take care. Bye bye.
You too. Bye bye.